So we've reached the last video for lecture 7, and this is, well, it's about bacterial quorum sensing, and I'll explain what that is. But this is something a lot of different bacteria do, and what you'll understand is that it's something that your own cells don't need to do. So this is something single-celled organisms have to do. It's very specific to them, and it's very clever. And one reason why we look at it is that it's more practice with understanding genetics and regulation. So we're going to see how some chemistry and some gene regulation lead to really complex and interesting behaviors. So this phenomenon is called quorum sensing, and that name comes from the fact that what bacteria are doing is figuring out if they have quorum. And so if you're on a committee, the committee meets and has to figure out if enough committee members are present to make a decision. If most of the committee members are not there, then you don't have what's called quorum, and that means you can't make a binding decision. That's a common rule of committees. And so that's exactly what the bacteria are trying to do, is figure out, are there enough of them to do something? So what sorts of things are they considering doing? Well, I'm going to show you two examples from two different bacteria. One of them, the first one, is from Vibrio fischeri, or Aliivibrio fischeri, and the Hawaiian bobtail squid. What happens here is that this squid, it goes out hunting at night for whatever a little squid eats. These things are about the size of a lime. And on moonlit nights, these squid would be visible from below, and fish would eat them. So what they do to get rid of their shadow or get rid of their silhouette when the sky is bright from moonlight and fish below them are looking up, they don't want that silhouette to show, right? They want to get rid of that silhouette. So what they do is they basically fill a specific organ they have with bioluminescent bacteria, and those bacteria make light. And the squid can control how much of that light gets out, and the squid can use that to camouflage itself. So that's what these bacteria are doing. And the squid encourages them to do that by feeding them. And so it lets some bacteria into its organ, feeds them, they grow, and once there are enough of them there, they can start making light. But this is an example of it's an example of a cool interaction in nature, a cool mutualistic relationship in ecology, but it's also an example of when quorum sensing is absolutely necessary. When Vibrio fischeri cells are alone, there's no point to making light, because a single cell's light is really faint and not detectable, and the squid wouldn't even notice it. So a single Vibrio fischeri cell needs to put its energy into dividing and making more cells. But a large group can stop dividing and instead start making light. And if they do this, the squid detects the light, feeds them, kind of rewards them, so to speak. So this is great, right? We can say, well, if a cell's by itself, it'll grow. If it's in a large group, it will instead make light. But how does the cell know whether it's by itself or in a group? And this is a complex thing that was not easy for bacteria to do. Bacteria can't see, they can't hear. All they can do is sense chemicals in water. And a big group of bacteria might be 10 centimeters wide, and a bacterium on one side of that has no way of really detecting a single cell 10 centimeters away. Bacteria are way too small to sense things far away, like a centimeter away. So what they do instead is use a quorum sensing system. So I'm going to walk you through this, and what you're seeing here again, is a small part of the genome of this bacterium. So this is Vibrio fischeri, and this is showing some of the DNA. And these arrows represent either genes or operons. So in this case, it's a gene, and the direction of the arrow shows sort of which way RNA polymerase is going to go when it transcribes the gene. This one goes in the opposite direction, and it's a big operon with 6 genes transcribed onto the same messenger RNA. And so if these get transcribed, if these become transcribed, what we get is certain proteins that are capable of making light. So they use ATP and oxygen in this complex chemical reaction that generates some light. So if the cell makes these proteins, if it transcribes this gene, this operon, and then translates these proteins, the cell will make light. What this one is doing is making a receptor protein. So this is making the LuxR, or Lux receptor. So, okay, interesting. What is this making? Well, that's making the autoinducer making protein. So that's making a protein that's going to make the autoinducer. And we'll get to that in a second. So we have sort of 3 different genetic elements we pay attention to. We think of all these 5 genes together as making light. We don't really care about any individual one. So we think if all of these are being transcribed, the cell will get light. This one, LuxI, the very first one in the operon, makes something different. It makes a protein that's going to make what we call the autoinducer. So this is going to make the autoinducer once this protein is made. And then this one, it gets transcribed at a different time in a different way. It's not part of the operon, so it's controlled in a different way. But when it gets transcribed and translated, it's going to make the receptor protein. There's another thing, and that's the DNA here. And this is the site where the Lux receptor can bind. And if the Lux receptor binds to this spot, it will basically increase the rate of transcription here. So this will be transcribed more often if the Lux receptor binds here. Well, what controls when this binds? The autoinducer molecule does. So this autoinducer is called an acyl homoserine lactone. So this is a homoserine lactone, and this is an acyl chain. So it's an acyl homoserine lactone, or AHL. And that's what the LuxI protein is making. So it just keeps making more and more copies of that molecule, and they can diffuse across the membrane. And that's what they do. They diffuse across the membrane. And if there's a lot of it outside the cell, like if other cells are there doing the same thing, some of it will diffuse back in, and the Lux receptor will attach to it, and that's what makes it decide to attach to the DNA and cause more transcription. So, again, here's what the parts do. This operon, if it's transcribed, is what leads to light production and creation of the autoinducer. This receptor, if it binds the autoinducer, it will bind the DNA here also, and it will cause more transcription of this operon. So, how does that lead to, well, yeah, this is the full diagram, sorry. So, how does that lead to a quorum sensing system? Well, let's think about it. One cell by itself is going to make the acyl homoserine lactone, and it will diffuse away from the cell. That's what's going to happen. It's going to keep making it, and it's going to keep diffusing away from the cell. And so the internal concentration will stay very low. And this is important, again, because one cell, if it made light, the squid wouldn't notice it, and the squid wouldn't feed it. So its job is to make lots of friends that can help it. If a cell has a bunch of friends nearby, what will happen is they're all making some of this acyl homoserine lactone, and it can't diffuse away. It just keeps bumping into cells. And some of those cells, it will go into them. And what will happen is that LuxR will bind it, and it will lead to more transcription of the whole Lux operon. And so we think of that as a positive feedback loop, because once this is transcribed and the LuxR binds the autoinducer, it's going to cause more creation of the autoinducer. So that's a positive feedback loop. Once this starts, it increases very quickly. All these cells, once they detect acyl homoserine lactone, they're going to make more very quickly. They're going to start making light, and they're also going to make more acyl homoserine lactone, and all of these cells will get activated and start making light. This is where the word autoinducer comes from. What it induces is more expression of the operon related to light production, but it induces itself. It induces its own formation. So it is an autoinducer. So that's how these cells control whether or not they do group behaviors. Remember, bacteria are smaller than neurons. They have no nervous systems. They have no logic systems other than genes and proteins. So this is how genes and proteins help them make that decision. Right, and quorum is achieved, light is produced. So this walks you through all of that again. So if I lost you, pause this and try reading through this and seeing if you can map this out. I would recommend drawing a diagram for yourself. And if you walk through that process of drawing a diagram, you have to make decisions about where you put things. That is a powerful way to learn this kind of thing. So that was Vibrio fischeri in its mutualism with the Hawaiian bobtail squid. And the cells were trying to decide whether or not to make light. In this case, the autoinducer molecule was a acyl homoserine lactone. And if we go back, I want to go back and look at that acyl homoserine lactone, which is somewhere back there. The acyl homoserine lactone has two parts that matter. This is the homoserine lactone, so it's made from a serine and a lactone. And this is the acyl chain. Well, it turns out that different species will make different versions of this that have different acyl chains. And so each one of those can have a different acyl structure, and so that way, each of those species, if it detects a different one from a different species, it will know, oh, that's not me. I'm not in a group of myself. I'm in a group with other cells, other species. And so by changing that acyl group, or by specifying that acyl group, they're able to distinguish themselves versus other species. In other cases, they make completely unrelated molecules. And a lot of different species use this strange molecule that has a boron atom in it. And species that use this will get confused about whether they're together with members of their species or a different species that also uses this. And that's okay. They've decided to be that way. So the way I think about it is any autoinducer and the receptor that detects it, they make a language that's specific to that species. So the autoinducer is the words and the receptor would be the ears. And so this allows cells to make complex regulatory networks. There are some species that have multiple quorum sensing systems that they can use to make a lot of decisions about what they do as a group. So let's look at the next example. And this example is going to be directly applicable to clinical sort of healthcare. This is about Staphylococcus aureus and what it's doing inside humans. So when Staphylococcus aureus is causing an infection in human tissue, if it's by itself, one cell, it has to do things like avoid going into the lymph, avoid causing inflammation, just stay safe and make new cells. That's what this is trying to do. And it does that by staying in one place. So it needs to express genes that cause adherence to our own cells. That's what Staphylococcus aureus will do when it's by itself or in a very small group. Once it's a large group, then it's going to attack the body. Then it's going to secrete toxins that are going to damage the tissues. It's going to secrete things that are going to slow down the immune response. It's going on the offensive. It's being aggressive. So instead of hiding, now it's going to attack. And it's going to do that by expressing genes that lead to secretion. So these are the two different states the Staph aureus will be in that are related to its quorum sensing system. And it has a system that works in the same way, but there's a fundamental difference that really changes what this looks like. So first, let's look at what is similar between these two systems. First, there's an operon, and when it is transcribed, it's going to make things that are related to the quorum sensing system. So it's going to make a transport protein. It's going to make a membrane-bound receptor. So this is the cell membrane. This is outside of the cell, inside. It's going to make this AGRD. This is the autoinducer. For Staphylococcus aureus, the autoinducer is a short peptide. So this is the gene that makes that short peptide. And so once these are all being made, the cell has an export protein that pumps this peptide out, a receptor protein that detects it if it builds up in the environment, and then one more thing that this AGRA is part of a phosphorylation scheme. So once the AGRC detects the autoinducer, it will phosphorylate AGRA to make AGRA phosphate, and that will act as a transcription factor that will change the cell's behavior. It will basically lead to transcription of what's called RNA3, and this is going to be a big regulator that's going to, by itself, do a lot of changes to the cell. So if this is being made, it's going to cause transcription of the secretion things, and it's going to block transcription of adhesion proteins. So, how is this similar to the quorum sensing system we saw before? Well, one of the main similarities is that one of the things that can be upregulated when there's a large group of cells is the same gene that makes the autoinducer. So the more cells of Staphylococcus aureus are present, the more of all of these things you're going to get, including the transport protein, the autoinducer, and the receptor protein in the membrane, and the phosphorylation protein. So the more cells are detected, the faster they detect more cells. And that is, again, a positive feedback loop. What's different between this and the Vibrio fischeri system is the autoinducer. A peptide cannot diffuse across a cell membrane. It is charged. It cannot cross a cell membrane by diffusion. By contrast, if we went back and looked at Vibrio fischeri, its autoinducer is a homoserine lactone, which can diffuse across the membrane. Well, that has big effects. The Vibrio fischeri didn't need a transport protein, and it didn't need a membrane-bound receptor, because its autoinducer would just diffuse out of the cell, and if it was concentrated, some of it would diffuse back into the cell. But with Staphylococcus aureus, its autoinducer is a peptide that has to be actively transported out of the cell, and it can't come back in, so instead the cell detects it with a receptor protein. So in this case, if there are a lot of Staphylococcus aureus cells, the autoinducer peptide is not going to build up inside the cell. It's going to build up outside and be detected by this receptor protein. So this is a different system. And again, I want you to walk through it and make sure you understand it and understand the difference and how One change in autoinducer from an acyl homoserine lactone to a peptide can completely change the way the system has to be set up. But in both cases, the autoinducer gets made, and once it's detected, more of it gets made faster and faster, and that's going to lead to faster detection. So in both systems, it's a positive feedback loop. So this is a lot of stuff to work through. Make sure you do so that you can explain this process. Again, think of Bloom's taxonomy. You want to get to at least the level of understanding so that you can explain how this works on an exam or quiz. So with that, this video is over, and I will see you later.